Greetings to you also. Previously, we looked at monohybrid inheritance. We looked at an introduction to Mendelian inheritance. That is what we previously looked at, and we looked at some of the definitions of Mendelian inheritance that we usually commonly use. We looked at terms like phenotype, genotype, homologous chromosome, homozygous state, heterozygous state. We went ahead and looked at terms like a testicles. We looked at a pure breeding parent. We previously looked at the laws of inheritance, where we said that we have a number of laws of inheritance, where we said that there are about basically three of them: the law of dominance, the law of independent random segregation or independent assortment. Now, we are going to go ahead and as well look at the Mendelian dihybrid inheritance today. After looking at Mendelian dihybrid inheritance, we are going to go ahead and also look at the non-Mendelian inheritance, because we have also what they call non-Mendelian inheritance. There are some aspects within our lives that are inherited in a non-Mendelian fashion, where we do not actually see the dominance law applying, or where we do not see the Mendelian ratio, which is called phenotypic ratio, which is 3:1, or the Mendelian phenotypic ratio, which is 9:3:1 apply. So in dihybrid inheritance, previously we looked at monohybrid, and we said in monohybrid inheritance, we are seeing breeding experiments that tackle inheritance of one trait or one character. That is to say, you focus, let's say, an example of length of the internode, and you only look at that. That is what we talked about previously. Or you focus on the color of the seed coat. That is what we focused on. And in the color of the seed coat, you look at maybe green and yellow. Okay? You don't look at the inheritance of length of the internodes. But in dihybrid inheritance, we basically look at both aspects. We look at how inheritance of color of the seed coat, along with maybe another aspect. Let's say an example of a trait like the texture of the seed of the pod, or we also look at it in terms of inheritance of the length of the internodes. In future, you will also interact, and you will find that we also have we have even trihybrid inheritance. So if we are looking at inheritance of two traits at a time, then we call that dihybrid inheritance. If there are three, trihybrid inheritance. If it is one, monohybrid inheritance. So in this dihybrid inheritance, that is where we get the law of independent assortment, where he says each of the pair of alleles segregate independently during gamete formation. That each of these alleles, remember, we are going to be talking about two different genes, but each of those genes, let's say the gene for color of the seed, the gene for color of the seed can exist in two different allelomorphic forms. That is to say, the green and the yellow. And let us now say the length of the internode. It can also exist in two allelomorphic forms. That is to say, tall and short. So when you look at those a pair of contrasting characters, those genes for the pair of contrasting characters are going to segregate amongst themselves independently. Okay? They are going to segregate among themselves independently. They are going to separate into the daughter cells independent of each other. That is the meaning of the independent assortment law. So the law of independent assortment is Mendel's major second law of inheritance, which talks about that each of the alleles within the genes of the pair of contrasting characters independently segregate during the process of gamete formation. And the formula that we use here is the 2 to power n, where n is the number of heterozygotes. So we are going to go ahead and look at a question. Here we are. How many gametes will be produced for the following allele arrangements? Okay. Remember, 2 to power n, and we said n equals to the number of heterozygotes. 2 to power n. Okay. Take an example of this. We have the first one, which is number 1. You would, if if you can pause this video and then you go ahead and look at the number of different combinations that we can have. Let's take an example of this one here. The first combination, we can have capital R, and capital Y. Then the second combination, we can have capital R and small y. The third combination we shall have is small r, capital Y, small r, small y. That is what we mean when we say that these alleles independently segregate themselves. So we shall end up with about 4 different combinations. These ones here. We shall have about 4 different combinations of the same. How do we arrive to that? We have 2 to power 2. 2 to power 2 equals to 4. So if we apply the same principle here, where we are having 2, now this 2 is going to be to the power of 4. So 2 to power 4, the combinations that we shall have are 2 to power 4, which will give us 16. Is it 16? 2 times 2 is 4. 4 times 2 is 16. And then 16 times 2, that is 32. The different combinations that we can form. So this one will automatically give us some other value. So, and the list continues like that. So we can see that the number we have seen that. We have seen this one here. We shall have 8 gametes, which is 2 to power 3. The last one we shall have. So you can see the different combinations. Then the last one. So these are the different combinations which are formed. So you can see. I will take an example of the first one here. You realize that you are seeing that we have here capital R independently segregated with capital Y. Then capital R, small y, small r, capital Y, small r, small y. That is the independent assortment in dihybrid inheritance. So these are gametes. 4 different combinations of gametes have been formed before fertilization can take place. Now, having looked at that, let us consider a dihybrid cross where we are seeing 2 traits. That is to say, we are looking at the shape of the seeds and the seed color. Let us assume that we have capital R, all round seeds, being dominant over wrinkled seeds. And let us assume the color, yellow color is dominant over green color. So round seed will take capital R, and then yellow, and then wrinkled will take small r. Capital R will be for yellow, and small r will be for green. When we have that, so our parental phenotype, if I was to write it, we shall have round yellow crossed with wrinkled green. Round yellow crossed with wrinkled green. But since now we have started with the heterozygote, now this one still will be round yellow, round yellow. Why do we say that this one will be round yellow and round yellow? Because capital R and capital Y are going to cover up the effect of the small r and small y. So this one will show the dominant characteristic. This genotype here will only be for the dominant characteristic and not the recessive characteristic. The recessive character will not be expressed. So we can have these are our gametes. So when during meiosis we form our gametes, and these will be the different combinations of the gametes in each of them. The good thing, they will be the same for each side. So those are the gametes that we have formed. Now, having formed these gametes that we have formed from the process known as meiosis, during meiosis we saw independent assortment taking place. Our next job is going to be all those are the possible different gametes that we can form for this pair of contrasting characters. Now, the dihybrid cross, we can now, the next step is going to go ahead. We are going to go ahead and look at the Punnett square that can help us to see the possible genotypes, of the offsprings, and the possible genotypic ratios and the phenotypic ratios of the offsprings. So we fill in the parental gametes. This is the male parental gametes, and then we fill in the female parental gametes up the female. Then on the side that is male. Then after doing that, we start fertilization. So during fertilization, we have filled in the gametes. That is how they are filled in. From yesterday, now you know how to fill in the gametes. This one goes with this to form this. This goes with this to form this. This goes with this to form this. This goes with this to form this. And when you're writing them, always remember that the capital letter, in case it is present, becomes the first letter as you're writing it, then followed by the small letter. In case you have the capital letter present, always make sure that you write the capital letter first before you write the small letter. So that is, those are our possible genotypes of our offsprings that we shall have formed. So when you look at the possible genotypes of our offsprings, if we are now told to go ahead and find out the genotypic ratio, or we usually use this to find the phenotypic ratio. This one here, what will this one be? This one here, what will be its phenotype?
It is round and yellow.
Round yellow. How about this one here?
Yeah. Still round yellow.
How about this one here?
That one is wrinkled and yellow.
And then this one here?
That one is wrinkled and green.
Wrinkled and green. So can we go ahead together and count the ones which are round yellow? Round yellow? You tell me.
1, 2,
yes.
3, 4, 5, 6, 7, 8, 9, 10.
Are you sure there are 10? Can we count together? Let's count together. Are they 10? Let's count. You say we have 1 here, we have 2, we have 3.
4, 3, 4, 5, 6,
7, 8, and 9. This,
there are 9.
yes, there are 9. So then we go ahead and count the ones which are they are yellow, they are round but green. How many are they? Round but green. So we look out for the capital letter of round of capital R and then small y, 2 small y's.
1, 2, 3.
There are 3. So there are 3. Then how about how many do we have which are wrinkled and yellow? Wrinkled and yellow. Let's count how many of them. So when we are choosing wrinkled and yellow, we shall look for small r, and then any capital Y.
1, 2, 3.
3. Then how about wrinkled and green?
I see 1.
1. That is how we arrive to this ratio, which is known as the Mendelian dihybrid inheritance ratio, the phenotypic ratio, F2 phenotypic ratio. That phenotypic ratio, which is labeled 9 to 3 to 3 to 1. That is what we call Mendelian dihybrid inheritance ratio, F2 ratio. Okay, now that takes us to the same thing here. That's how they look like. You can see how the yellow look like round yellow, and so on and so forth. That is how they look like. Then we can go ahead and look at our test cross, how we can establish our test cross. Remember, we talked about previously, in our previous video, we talked about a test cross, and we said the test cross is usually carried out to ascertain the genotype of a suspect who is expressing the dominant gene. He's expressing the dominant gene. So we want to find out, is this individual homozygous or heterozygous for that specific dominant gene? Because the recessive one is always clear. We said the recessive gene only expresses itself in homozygous state and not in heterozygous state. Okay, so that is the question that we usually have. That is why here, in this question that we are having, in this example, we are having this one here, who is double recessive, and this one whose genotype is unknown. Whether here, in this position here, which is where we have the dash, is a capital C or a small c. We want to find out. Okay, so now, let us assume capital B is brown eye, then capital B, small b is brown eye. What does that tell us? That tells us that the gene for brown eye is dominant over the gene for the other eye, which is maybe probably blue eye. Okay, so small b is blue eye. I've already explained this very well, that now From when you look at this one, capital B, and then capital B, small b, giving you brown, then that is enough to tell you that this gene for brown eyes is dominant over the gene for blue eyes. Why? Because it can phenotypically express itself in both the homozygous state and the heterozygous state. Now, and then the other one is curly hair, and then you can clearly see now of these two, curly hair and the of the two, curly hair and straight hair, which one is dominant over the other? Over the two, curly hair and the straight hair, which of them? Curly hair is dominant. Thank you. So now, having noticed that, now when we are carrying out a test cross, we want to ascertain the possible different possible results. So we can have a possible result of this or possible results of this when we carry out a test cross. When we want to find out, a test cross is usually carried out to ascertain the possible genotype of an organism that is exhibiting a dominant gene. Okay. Now, a summary of Mendelian laws. That's a summary. This slide is trying to summarize all the Mendelian laws. The first law, we said dominance cross, where we said that in the parent cross, when we have capital T crossed with small t, we shall end up all the offsprings showing the because the offsprings are hybrids, then one of the genes will dominate over the other, and the one that dominates over the other is known as the dominant gene. The other one which is dominated over is the recessive gene. Then the second one is segregation. In segregation, we said these genes, these alleles, yes, a characteristic is controlled by a pair of factors, but each of those two factors tend to segregate each other. They tend to, each of the gametes tend to contain only one copy of each factor. That is the segregation. And that is when we get the heterozygotes and we cross them, we end up with the we end up with 75% of the dominant one and 25% of the recessive one. That is what we said when we looked at the law of segregation. Then when we looked at the law of independent assortment, we said in the law of independent assortment, we look at the genes, where we said if we are to consider a pair of contrasting characters, take an example, two genes, 4 different characters. They independently assort themselves, and then we end up with a ratio which is to say 9 to 3 to 3 to 1 phenotypic ratio. That is produced as a result of independent assortment. We said that they combine with each other independently. Okay. Now, having looked at that, now we are going to go ahead and look at some of the deviations. These inheritance, there are some characteristics whose inheritance does not abide with the Mendelian laws as we have stipulated them above. So that's what we are going to go ahead and look at. There are some inheritance, certain characters that each are inherited and they do not follow the Mendelian inheritance fashion. And those are the ones we are going to look at. We are going to start with what they call incomplete dominance. Some characters are inherited in a fashion, in a way that one of the characters incompletely dominates over the other. And when we get up, we end up with a phenotype which would actually either be tall or short, according to Mendel. But we end up having an organism with intermediate characteristic. Then that one is incomplete dominance, intermediate height. Okay, so we are going to start with incomplete dominance. Now, in incomplete dominance, the F1 hybrid has appearance somewhat in between the two phenotypes, and it is more to a given character. It is more to a given character than the other. That is why they say incomplete dominance. One of the genes tries to incompletely dominate over the other. Take an example, as we are going to see. So the parents, let's say we have parents. Let's say I'm just using height. In case you have a tall, homozygous tall and then homozygous short, when you cross them, according to Mendel, you expect to have the hybrid being either very tall or very short. But in incomplete dominance, you will have the hybrid of intermediate height. let's take an example. An example is the snapdragon flower. When you look at the snapdragon flower, when they crossed the red flowers with the white flowers, homozygous red, homozygous white, they ended up with the red, usually, unfortunately, yes, we might use capital letter, small letter, but in some textbooks you'll find that they are saying in incomplete dominance we have capital R and capital R for red flowers and then capital W for white flowers in some textbooks, and that is the one which is most widely accepted as opposite to capital R, small r. This is Mendelian, okay? So the most correct thing we would have here is W, because each of them is going to express itself, but of course one is dominating over the other when you look at this snapdragon flower. So when we have these ones here and we cross them, we have capital R on the male, and then small r, or what we have agreed as capital W. You as the gametes that we have, you end up with, let's say, you end up with the capital R, small r as your phenotype, as your genotypes, your F1 genotype. Now, when you look at the phenotypes, you'll realize that they appear to be pink. Instead of either being red or white, they are pink. Pink is an intermediate color between red and white, but it is more to which color? Is it more to red or more to white?
It's more to red.
Good. That means that the gene for red color was trying to dominate over white, but it was incompletely dominating. That is why they call it incomplete dominance. Okay, so this one here is an example of incomplete dominance. There are other examples that you interact with. So this is an example that we have seen: the pink flower, the red flowers, and the white flowers. When you cross both of them, you will end up with offsprings which are pink. So when you get the pink flowers, and then you allow them to crossbreed with each other, self-breeding, and you get the F2 generation, you realize that you will have, among your offsprings, you will have white flowers. You will have when you get these pink flowers and you cross them, among your offsprings, you will have one white flower, two pink flowers, and one red flower. So you will get a ratio of 1 to 2 to 1. Are you seeing this? 1 to 2 to 1 is deviating from the other ratio that we had talked about earlier, which is supposed to be 3 to 1 F2 phenotypic ratio. We expect a 3 to 1 F2 phenotypic ratio, but here we are having 1 to 2 to 1. That shows us that now these genes are against the Mendelian inheritance law, which says with the dominance law, they are against dominance. So we end up with a 1 to 2 to 1 ratio. Okay, that is sorted now. That is incomplete dominance. Now we go ahead and look at the other phenomena known as codominance. Now in codominance, for it is unique. In codominance, two alleles are expressed in the heterozygote, and they're expressed equally. These alleles, they try to express themselves equally, with none of them dominating the other. Like previously in incomplete dominance, we saw that the allele for red flowers was trying to dominate over the allele for white flowers. Now, in codominance, all these alleles try to express themselves equally in the phenotype of the heterozygote or the hybrid, and that is why we call it in codominance. We have very many examples of codominance, like during inheritance of fur color. When you look at inheritance of fur color, when you look at the cows, when you get a black colored fur cow and you cross it with a white colored cow, you will end up with a cow which has equally distributed white fur and black fur equally distributed on the hide. We have very many others, and that cow is called the lone cow. You have heard about those cows. Red and white still, and then you get a calf which is made up of both red and white fur on its hide, and they are equally distributed. That one is called a lone cow. In human beings, we have this one example known as blood groups. Blood groups are an example of another phenomenon known as multiple alleles. You see, now tomorrow they will ask you to explain different deviations from Mendelian inheritance. I would like to inform you that when you look at multiple alleles is another small deviation, which is even different from codominance. So you can give it as a different deviation from Mendelian inheritance, though we are going to look at it in details as an example of codominance. In blood groups, you will realize that we have the different blood groups according to the different antigens that are found on the surfaces of the red blood cells that we possess. Individuals who have blood group, so you can see here, a person who is of blood group A, type A, has antigens which can either be capital A, or capital A, and capital A and O. This one here, small I, is O. And this bar that we are drawing here, which looks like a capital R, capital I, this one represents immunoglobulin. Immunoglobulin is a word that is usually representing the antibodies, but we just use it. It's a symbol that is showing us that this thing is related somewhat to blood. But anyway, either way, they are all correct, even if you don't draw it and you just draw capital A, capital R, capital A, or capital A and O. Because usually in multiple alleles inheritance of blood color, we have the ABO blood group system. It is very important to put it down, note it down in your notebook, and say that in codominance, the gene for allele A and B are codominant to each other, meaning that when those two alleles exist together in the heterozygote, they equally express themselves, and that is why that individual will be of blood group AB. Then, when you look at that is an example that we are having here. Then, when you look at the allele for blood group A and the allele for blood group B, those two alleles are dominant over the allele for blood group O. That is why this individual here appears to be of blood group B, because he has one of his alleles as capital B, and in the absence of the other allele, which is O, now this person will appear to be blood group O, blood group B. Then this one will appear to be blood group A. This one will be AB. Because the allele for blood group A and blood group B are dominant over the allele for blood group O. And then the allele for blood group A and blood group B are codominant to each other. That means that the allele for blood group A and blood group B, they can equally express themselves when they are present. That is why the person will end up with blood group AB. Now, in multiple alleles, a character is controlled by genes which exist in more than two allelomorphic forms. An example is this one here. The different allelomorphic forms, we have A, we have B and O, but at every given point, we only have two existing. You can never have all the three existing together, because when you look at the chromosome, where they are found, the area known as the loci can only accommodate two at an interval. So that is why we have the different combinations which are in the twos. So that's how you end up with this. So that is codominance. Now let us go ahead, and so blood group O will only exist when both alleles when both A and B are absent. When both A and B are absent, then we end up with blood group O. When either of the two is present, you will end up with either A or B. When both of them are present, you will end up with AB. When one of them is present, you still end up with A or B, respectively, depending on the type of allele present. Now, codominance problem. Let us look at a codominance problem. This is very important because it will enable us to solve certain paternity disputes. Before you run to DNA test, like it is very famous of nowadays, people are testing DNA so much to ascertain the paternity of the child. A long time ago, when they used to settle paternity disputes in courts, they used to use blood grouping to try to find out whether this is your child or not your child, though it has its own inconsistencies. Sorry. So now we can go ahead and look at this problem here. We have a parent, two parents, a female and a male. This male here is homozygous for blood group B. When he's homozygous for blood group B, that means has both alleles as B. When he's heterozygous, like the mother, he has one as B and the other one is O. So when we try to cross these two, you will end up with something which looks like this. You will end up, these are your offsprings. So you will have this cross with this, then that will give us blood group AB. Then this with this will give you blood group AB. Then still, now you will have this with this, they will give you blood group B. Then this with this will give you blood group B. So something like this. So when you look at your ratio, because this one is blood group B, this is blood group B. Why is it blood group B? Because B is present and B is dominant over blood group O. This one is blood group AB simply because the allele for blood group A is codominant with the allele for blood group B. So that is why this person will be AB. Okay? This person here will be AB. Then this one will be AB, this one will be B, this one will be B. That is how we arrive to that. Now, having looked at codominance, we can look at another problem, another codominance problem. Take an example. We have this. We have these two. We have the male is the blood group O and the female is blood group B, AB. Can we go ahead and calculate? Now, this one here, you can pause and then you calculate it. I'm giving you 2 minutes to calculate this one. So now, let's look at, I hope you have got the correct answer. So let's go ahead and see our solution. Okay, there we are, our solution. Let's go ahead and see. So you can see, those are our genotypes, the possible genotypes. Then we have, that is the ratio. I hope all of you have got that ratio. Now, that is the ratio that we have, half of blood group A and half of blood group B. Why are we saying so? Because the allele for blood group A is dominant over the blood group O, and the allele for blood group B is also dominant over blood group O. But allele for blood group A and B are codominant to each other. Okay, so there we are. That is codominance. Let us go ahead and try out another exam, another question, and I will also give you 2 minutes to answer that question. So let us go ahead and see our answer, whether our answer that we have got is correct. So we have now, we have here, in case we have the parent of blood group O, then the parent of blood group AB, the boy, then the girl. The boy, remember, if a boy has blood group O and his sister has blood group AB, those are children, offsprings of two parents. So we want to find out the genotypes and the phenotypes of their parents. Okay, let's go ahead and find out. Okay, the answer is, when you look at now, how do we ascertain that we now do we are now doing backwards? Which genotypic genotypic combination gives us the parents? You can see, in case we have this, we can have the parents having their genotypes as blood group A and blood group blood group A and then blood group B. Those two parents can give us a child of blood group O and a blood group AB child. How? Why? Because this one here, who is of blood group A, is heterozygous for blood group A, and then the father is also heterozygous for blood group B. So since they're heterozygous, they have a silent O here and a O here. So when you cross, you will end up with a blood group. This is blood group O and this is blood group AB. These are the two people who were produced from this cross of these two parents. That is codominance. Okay, so the next deviation from Mendelian inheritance that we can talk about is called sex linkage. Now, in sex linkage here we look at a number of issues. Now, in sex linkage is broad, or it is under what we generally call linkages. And the linked genes are genes which exist together on the same chromosome and inherited together. That is linked genes. They exist together in a linkage group and they are inherited together. That is linked genes, or linked. The traits are the ones whose genes are carried together on the same chromosome and inherited together. So specifically, we have two types of linkages. We have sex linkages and autosomal linkages. You will come across autosomal linkages, but today we are looking at sex linkages. In sex linkage, we usually see traits whose genes are located on the sex chromosome and have nothing to do with sex determination. They are carried on the sex chromosome but have nothing to do with sex determination. But they are inherited together with those genes which control sex. So they are carried on the sex chromosome and have nothing to do with sex determination. That is to say, they are carried on the X chromosome and the Y chromosome. But the most common ones are the X-linked genes, not the Y-linked genes. Basically, the most common one is the X-linked, but not the Y-linked. Okay, so females usually have a genotype of XX, and males usually have XY. So many sex-linked traits are carried on the X chromosome and not the Y chromosome, because the Y chromosome is usually considered to be genetically empty, though it can carry some of the genes for certain characteristics that are only unique to males. And those characteristics that are only unique to males are known as halondroic characteristics. Halondroic characteristics, HALONDROIC, halondroic characteristics. They are characteristics which are only found in males, like hairy ears. Now, sex-linked traits, we can give an example like in the fruit fly. The red eye color in fruit fly is sex-linked, and it is carried on the sex chromosome, specifically the X chromosome. So you can see the X chromosome is usually big in size, and it carries those genes. The Y chromosome is genetically small in size, so that's why they say it is always genetically empty. But it is misleading. That statement is very misleading. It is not empty, because it can sometimes carry the halondroic characteristics. Okay, let's look at an example here where we are seeing red-eyed male crossed with a white-eyed male. So let us assume, let's take maybe let's say red eye is dominant over white eye. Yes, red eye is dominant over white eye. But also remember, but they are carried on the same sex chromosome. So that is why we have capital R being red-eyed, and capital R small I being small r capital R being red-eyed, R small being white-eyed. Always remember to define your symbols that you are using. In case you do not define your symbols, the examiner will not know the meaning of the symbols that you are using. That is why we endeavor for every cross to say what R represents, what capital R small r represents, what XY represent, and what XX represents. Always remember to define your symbols that you are using, because these are simply representations. They are not letters. They are symbols representing something. Okay, there we are. So we can go ahead and see this cross here where we have a red-eyed male and a white-eyed female. Okay. There was an error, but now this is correct. This is better. Sorry, there was a typing error where we had small r here. White-eyed female will be small r, and the red-eyed male will be capital R. Remember, the Y chromosome is genetically what? Empty. That one you should remember very well, that the Y chromosome is genetically empty. So you can see here and see what we are going to get. Okay, we shall have, those are our parental genotypes. Those are our offsprings, the offspring genotype. You can see we have now these ones here. We have capital R, small r, capital R, small r, and you can see here. So now this one here will be red-eyed, but white-eyed what? Males. These ones will be white-eyed males. This one is going to be red-eyed females. Okay, let's see our ratio. We shall have 50% of the females being red-eyed and 50% of the offsprings being white-eyed males. So you can see there is something unique here that we need to understand. When you looked at sex-linked characters, so this is deviating from Mendelian cross, which says 3 to 1. Now this is 1 to 1 ratio. This is deviation from Mendelian inheritance. Mendelian inheritance was telling us 3 to 1. This one is 1 to 1, which is wrong. Now it is against. Now, remember, you can see that the red-eyed character was in the males, but it has now been carried to the females. Are you seeing that? This is very important for us to remember. Remember that sex-linked characters, if they are to express themselves, let us say, there are other examples, like in human beings, we have the red-green color blindness. For a girl to be colorblind, that means that it was not his mother who was the problem. The problem was the father. If the boy is the one who is colorblind, it is not the father, but it is the mother, because of the way they are inherited. You can see clearly here that now the mother was having white eyes, and now they have gone to the male children that she will have. Okay, that is done. So another thing that we need to note about sex linkages, we need to note very well, and we need to note and say they are more pronounced in males than in females. Females, most of them are carriers. Females, most of them are carriers. But for it to, why is it that they are more common to males than in females? Simply because the males have only one X chromosome. So that X chromosome should either have the normal gene, which is capital R, or the recessive gene, which is small r. But it cannot have both of them. So that is sex linkage. So they are more common in males. Why? Because in females, for them, in case the defective gene is present on one homologous chromosome, on one X, the normal gene will cover up and mask the negative effect of this other gene. And that is why you will end up with females being carriers. So